Sickle cell disease is a group of inherited disorder that affects the red blood cells. The function of RBC is to carry oxygen from the lungs to the rest of the body. To perform this vital task, RBCs are filled with hemoglobin protein. These protein float independently inside the RBC cell, keeping the cell flexible enough to squeeze through even the tiniest of the blood vessel. Sickle cell disease is caused due to a genetic mutation that alters the structure of the oxygen-carrying protein hemoglobin to deform it into long pointed sickle. These RBCs are harder and stickier and does not flow smoothly through the blood vessels. After releasing oxygen to tissues, these mutated proteins lock together into rigid rows. Sickle cells snag and pile up, sometimes blocking the vessel completely. This deprives a variety of cells from oxygen, causing wide range of symptoms experienced by people with sickle cell disease. Patient suffers from repeated episodes of stabbing pain in oxygen-starved tissues. The location of the starved vessel determines the symptom. A blockage in the spleen puts patient at a risk of infections. A pileup in the lungs can produce fever and difficulty in breathing. A clog near the eye can cause vision problem and retinal detachment. And if the obstructed vessels supply the brain, the patient could suffer a stroke. While a healthy red blood cell survives for 4 months, sickle cell survives for just 10 to 20 days. This short lifespan means the patient lives with a constant depleted supply of red blood cells, leading to a condition called as sickle cell anemia. Sickle cell anemia developed as a genetic mutation for a tropical disease called malaria spread by Anopheles mosquito. Malaria uses the RBCs as incubators to spread through the bloodstream. The structural changes brought in the RBCs made them less resistant to malaria. In places with malaria, sickle cell mutation offered an evolutionary advantage. For a baby to be born with sickle cell anemia, both parents must carry the sickle cell gene.